Okay, well, hello, everybody. Thanks for joining us today. We are going to talk about cancer detection using an ELISA lab. This is a lab that is super versatile. We'll talk about different ways that you can use it, but we are going to be focusing on how to use it to detect cancer biomarkers today. My name is Lee Brown. I'm a curriculum and training specialist for Bio-Rad Laboratories, and Tamika Stubbs is helping me in the chat. So she'll be there to answer any questions you might have. If you want to just type those into the chat, she can either interrupt me if it's, something really important that we need to go into right away, or we can save them to the end. Either way, we'll have time at the end for, I think, for some Q&A as well. And also, I wanted to make sure, so here's where we are. So I cover kind of the central and southeast US. Tamika covers the northeast, east coast, and then we have two more colleagues who cover the western and central part of the country. And really, any of us can help you with any of your questions. We're just here to kind of help make life easier in your classroom. So if you have questions about this lab or other labs, equipment, how you might integrate some of these biotech techniques into your health science or your biology class or a full-on biotech class, we are here and happy to help you with any of that. We have today a very special gift, I guess I could say, for at least one lucky person. We are going to be giving away a free kit for actually every 25 participants. So we might be giving away 2 free kits. So if you wouldn't mind hitting this QR code with your phone and just entering in your name, all we need it for is just to have a list of participants so that Tamika at the end of this will pick a random number and we will draw a name from that. So you don't have to give us any other information, just your name so that we can have that in our list here. I'll leave that up for just a second, let y'all do that. And Tamika also has a URL, so she can put that into the chat as well once I navigate away from this slide. Oop. Okay, and I'm giving you this slide deck here. I'll be sending this out with the recording probably, let's see, it's Thursday, probably Monday. We're trying to get these up on YouTube. And so I'm hoping to send you the link to that. I'll send you the slide deck as well, just so you know this is kind of organized with different color coding. So the yellow stuff is just kind of resources to help you teach with, teach this with your students. The green is all the background information. That's really what we're going to focus on today. There's a section for instructor prep. I probably won't spend a lot of time on that. We'll go over the student protocol a bit, and we'll also take a look at some results. But just know there's a lot more information. I'm not going to cover all of that today in this presentation because it's just a lot to do, right? So I'm going to start off with some of the educational resources. We are using our biorad ELISA kit. This is a kit that covers 48 students, so that's really nice. You can actually even cover more if you're not doing things in triplicate like we're going to be showing here. But it's super versatile. We're going to be using it in a quantitative setting to detect cancer biomarkers and see, how much of them there are. But you can use this for really anything. If you wanted to look at HIV or COVID, we've even done this with like the zombie apocalypse. You can just make up a scenario if that's something that you wanted to do with younger students or students that aren't necessarily super focused on health science or biotech, right? So that's always fun. It comes with everything you need to do the lab, all the consumables. This one can be a no equipment lab. I will tell you more about that once we get to the protocol. We have some classroom resources here as well, and of course a textbook that goes into any of this. If you're thinking about starting a biotech program, or you just want to take a look at that textbook, just let us know and we can see what we can get to you. All of the protocols and a lot of the background information, though not the cancer part, are available in our in the manual, and this is a free download. So again, we will give you this PowerPoint so you can just download it straight from here. We also have a paper model. This has cutouts of antibodies, antigens, substrate, wash buffer, all of that stuff. I'm not going to go into detail on how to use that here, but we do have a whole PowerPoint, and I think we have even a webinar explaining how to use this. So feel free to use this alongside your regular ELISA. It just gives students a nice way to manipulate and see how things go together, how antibodies and antigens bind. We'll kind of talk about that as we go here. So I'm going to go into the background information here, and some of this background information is on cancer, some of it's on ELISA. So we're going to start off talking about how we might detect cancer to begin with. What are cancer biomarkers? How can we understand the cancer cell cycle and use molecular diagnostic techniques to detect cancers at an early stage, especially. After that, we'll talk about more about ELISA, what that is and how that how we use antibodies and antigens to detect different proteins, either in our serum or in, this could be a drug test, it could be a urine sample, all kinds of different samples, spit sample. We've all done that with COVID, right? We'll talk about different ways that you can use ELISAs, and then we'll go into kind of the nitty-gritties of how the protocol works, and I'll go over again the paper model real quick, okay? So let's start off with this cancer biomarker detection. Now, so as you probably know, cells typically will divide to make two identical daughter cells, right? And they can stop dividing and actually differentiate. That's when they're going to specialize in structure and function. That's how we get things like neurons and muscle cells and eye cells and all that, right? The third option, so we have division, differentiation. The third D here is they can die. So cells can undergo kind of this programmed cell death that we call apoptosis. And that is a good thing. We always think of cell death as being maybe bad. No, we want cell death to happen sometimes, especially when our cells are damaged, when that DNA is damaged and not non-repairable. Also, this happens during development when we're having cells that are, between our fingers, for example, some of those cells will die off. So apoptosis is just a normal thing that happens, and it's really necessary during development and also during all the other stages of life too, right? So now when we're looking at cancer, what is actually going on there? So if we think about cancer and some of the signs of cancer or symptoms of cancer, some of these will be familiar for you and some of them won't. But if we're looking at a molecular level, what's happening, right? So we know when someone gets cancer, we might see a tumor, or there might be the presence of a tumor, and also there will be, all kinds of symptoms, right? They're fatigue, things just aren't working right. Many different, cancer is hundreds of different diseases, so you can't just say there's this one set of symptoms that's true for every cancer, but a myriad of symptoms. But on a molecular level, if we look inside our cells at what's going on, let's take a look at what might be going on there. So there are some hallmarks of most cancers here. So we have kind of this sustained proliferative signaling. This means that basically our cells are saying everything's a go, like go, 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 make more DNA, we're all good to go here, right? That is not a great thing. We want to make sure our growth is really regulated and that cells aren't reproducing and making more copies of themselves unless everything's okay. There's also this evasion of growth suppressors. So we have growth suppressors that are always active, and cancer cells find a way to get around that, right? They tend to be immortal, so we don't have that apoptosis. So instead, cells, even though they're damaged or exhibiting some weirdo, effects or signs, they are still not dying, right? There's not that programmed cell death isn't happening. On top of that, and I'm going to stick on the left side here. So these original highlights. So metastasis is when cells that are cancerous may break off and travel to other parts of our body and form another tumor there, right? So that process is turned on. They're more likely to invade other tissues and to break off and go to other places in our body. They do that partially by forming more blood vessels, right? So this inducing angiogenesis, that helps them move into other tissues. And of course, this resisting cell death. Now, there's some other things that we've identified as kind of other hallmarks to look for with cancer cells. And a big one is avoiding our own immune system, right? So our immune system is really pretty good at detecting when things go wrong and, wiping that out. But cancer cells are always co-evolving with us, right? And so they have figured out ways around the immune system to evade the immune system, to keep them from noticing that that's a cancer cell, right? So there's all these other things that are going on with cancer cells, and this is just a few of them. What I really want to go on to now is taking a look at the cell cycle. So after a cell divides, it's going to go through a series of steps, right? So we have growing and replicating the DNA, preparing to divide, and then dividing. And that whole process here is called the cell cycle. And its main parts are known as phases. And so in interphase, we have, interphase is kind of this whole section right here, G1, S, and G2. That's going to take up most of the cell cycle. That's where our cells spend most of their time. In G1, the cell is growing, getting bigger, performing its normal functions, right? And they will also then move into the S phase if everything looks okay, if the DNA is in good shape and all that. In the S phase, DNA is replicated. Okay, we need extra copies of DNA here, right? Obviously, in G2, we're going to go into a little bit more growth and prepping for mitosis when the cell is actually going to divide. Now, there is this thing right here called G0 where it can kick out and go into a cell that's being differentiated, right? And so just doing its normal stuff and not going through the cell cycle of preparing to divide. Okay, now then we have mitosis, right? And so we go through prophase, metaphase, anaphase, and telophase. I'm not going to go into detail on all these, but this is something that you probably talk about with your students if you're doing a biology or molecular biology course, right? Okay, so here's our cell cycle. Now, at each of these phases, or in most of these phases, there are what we call checkpoints. And a checkpoint is when the cell kind of takes a look at itself, does some internal reflection here, right? And decides, is everything a go? Like, is everything looking okay? There's no abnormal weirdness going on. DNA looks good. It's not damaged. The cell's big enough to divide. We don't want to, pinch a cell in two if there's not enough stuff to go around, right? There's not enough proteins to go around. After the G2 checkpoint, we want to make sure that the DNA replication went okay when it's planned, and there's still no errors or damage. After that, if there are errors or damage and they can't be fixed, that's when we have apoptosis. There's also an M checkpoint in mitosis where we want to make sure that the sister chromatids are correctly attached to the microtubule, the spindle microtubules, right? And so all these checkpoints are there to prevent things like cancer from happening. These are checks on cell growth that are the normal part of our cell cycle. Okay, and so when errors occur in these checkpoints, this is really strictly regulated, right? And so if there's errors that occur there, there can be really catastrophic consequences, including the development of cancer. Okay, so with errors, there is often some DNA damage. Now, there could be, I don't have a slide on, mutations involved in all the cancers here, but a mutation can be inherited, but also we can have DNA damage happening, at any time, right, from a number of different things, environmental, things you eat, things you're exposed to, right? And so there's all these different ways that DNA can be damaged. So you might have something happening during replication. There might be these radicals or radiation. Chemo itself tends to cause cancer sometimes, right? So we're interfering with the growth cycle or with the cell cycle of quickly dividing cells that can sometimes cause cancer or other problems in, okay or normal tissue as well. UV light, of course, causes cancer, all these things. And so we can have these different sorts of DNA damage where we have a base mismatch or a single strand or double stranded break, right? And so that will throw everything off and have downstream effects, when we go to transcribe and translate the gene that DNA damage occurred in, okay? Oops, let me turn that all on. There we go. Okay, so now let's talk a little bit about tumor suppressor genes and proto-oncogenes. These are two sort of classes of genes, and they encode proteins. I'm going to start off with the tumor suppressor genes first. So tumor suppressor genes are genes that tend to be often involved in the cell cycle, and they are kind of like the brakes. They're going to inhibit cell growth and proliferation and hopefully the progression of tumor formation, right? And so these are often called the guardians of the genome because they regulate that cell cycle progression. And so they are protective against cancer when they're functioning properly. So they prevent the development of cancer by regulating that cell cycle progression, by regulating DNA repair and apoptosis. And so if those are inactivated or they lose their function of those tumor suppressor genes, again, that would occur through mutations like we just saw earlier, deletions, might be epigenetic silencing, right? So we're learning more about that now, of course. That might lead to the loss of their tumor suppressive activities. So this is like when the brakes in your car go out, right? So basically, now the cell is free to cut loose and we don't have the brakes going, wait, hold up, is everything okay before we divide? Okay, so there's lots of examples of tumor suppressor genes here. P53, retinoblastoma protein, P10, BRCA1 and 2. Those are breast cancer susceptibility genes. You've probably heard of a bunch of these, right? If you're at all interested in cancer or have looked at this, P53 is one of the big ones. And so any kind of mutation in those may lead to cancer, okay? Now, if we're looking at proto-oncogenes, those are normal cellular genes that typically, when they're all working fine, they're going to regulate cell growth, proliferation, and differentiation. When those are mutated, those proto-oncogenes become oncogenes, okay? And that's when they start promoting this abnormal cell growth and kind of this uncontrolled cell growth. They inhibit apoptosis, so we don't have that programmed cell death. And they're going to be driving this formation of tumors, okay? There are, again, more examples of this. RAS, HER2 is one we're going to look at today. This is, an overexpression of the HER2 gene, also known as ERBB2, and that's in breast cancers and also gastric cancers as well. And let me show you what that looks like. And then EGFR, epidermal growth factor receptor. Okay. So if we're looking at those two in context here, right? So with a normal cell division, here's our cell cycle right here. A normal tumor suppressor gene is going to act again to put the brakes on things, and a proto-oncogene is going to be telling it to go, right? And those are working in concert to progress cells through that cell cycle in a way that is controlled, but is also careful to look for all of these different things at those checkpoints. When you have malignant cell division, you can mutate this tumor suppressor gene or mutate a proto-oncogene. And when a tumor suppressor gene is mutated, we lose that function. So we lose the brakes on the car, right? And so that means that cell cycle is just going. With a mutated proto-oncogene, it's kind of I don't want to say the opposite. It's the other side of that coin, right? And so this is like having a gas pedal that's stuck down, right? Instead of our brakes failing, now our gas pedal, our accelerator is always on. So it's a gain of function mutation. And so again, we're just proceeding through that cell cycle without any kind of stops on it, okay? Mutations in proto-oncogenes, they cause this gain of function. You only need one allele mutated to have the situation, okay? Tumor suppressor mutations, those result in a loss of function, right? So those are recessive, okay? So you would need to have two mutations in order for those to cause problems, okay? Now, I'm not going to go into all of this about breast cancer, but this is a really nice figure. These figures came from a place called Bio-Rad. We have a subscription to that. It's a super cool, if you haven't heard of it, Tamika can write the Bio-Rad website in the chat there, but they have really, really great editable images. And so that's what a lot of these came from. So breast cancer subtypes can be categorized based on their expression of these different hormone receptors, right? And so there are kind of these 5 main molecular subtypes of breast cancer. The ones down here, TRP-negative ones, are the worst prognosis, right? HER2 is the one that we're going to be looking at today, and that's, again, a receptor. And when we see an overexpression of that receptor, that tends to be a worse prognosis. It's a more aggressive type of cancer, and it grows faster, okay? So that's the one that we're going to be focusing on today. I think we kind of covered, I'm going to come back to that. So let's look at what HER2 does in breast cancer. So HER2 is human epidermal growth factor receptor 2. It's a gene that encodes a transmembrane tyrosine kinase receptor. Okay, and it's part of this EGFR family. So let's take a look at what happens when it's normally expressed and when it's overexpressed. Okay, so here on the left, we have a normal breast, well, I should say a normal cell, right? So in normal cells, HER2 is involved in those signaling pathways that regulate cell growth and survival and differentiation. And when a growth factor binds to HER2, it's going to activate this intracellular signaling pathway to promote the cell proliferating, okay? And we want that, right? We want cells to grow and divide in a normal situation. Now, what happens with breast cancer, and this is in about 20 to 30% of breast cancers, we have an overexpression of HER2. So we have this protein that's being overexpressed, right? That gene is actually amplified. So we're getting more copies of the protein, this transmembrane protein. And that means there's, all these extra copies here. And so we have extra HER2 protein expressed on the cell surface of those cancer cells. Now, what does that do? That is going to lead to a kind of this continuous activation of those signaling pathways. They're always turned on, right? And that's even in the absence of any kind of external growth signals that would normally bind on the outside here. These are just always turning on. And so we're always getting, we see these arrows here pointing to the nucleus where the transcription of these genes is happening. So that is just this constant input telling those to go, 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 right? Make more and more and more copies, okay? And again, that tends to be a much more aggressive form of cancer. Okay, just as a side note here, Herceptin, which is a chemotherapy, it is actually a monoclonal antibody, and it works by binding and kind of cross-linking these Herceptin protein, right? And so it blocks the activity. So that's a chemotherapy for HER2 positive breast cancers. Obviously, this won't work as well, or I don't think they use it at all if it's not, if we don't see that type of breast cancer, okay? Whoops, here we go. So here's that internal signaling pathway here. I'm not going to go into the detail. I couldn't even tell you all these details, but you can see here that we have this, these growth factors here. And when they bind, then HER2 causes a signaling cascade inside, and we get these genes being turned on that are important for cell proliferation, cell survival, and progressing through the cell cycle. Okay. Now, there are other cancer biomarkers that you could also look at. I picked HER2 just as a, one that's involved in breast cancer and also gastric cancers, but PSA is another one that we often look at with an ELISA, and that is associated with prostate cancer. Alpha fetoprotein is associated with liver cancer. This EGFR, epidermal growth factor receptor, is similar to HER2, so another type of growth factor receptor with various cancers. And so there are lots of other cancer biomarkers that you can look for. And what we're looking for is something in the serum of a patient to see, are we looking at something that's expressed at a normal level, or is it maybe expressed at a higher level, right? And so that's what we're going to use an ELISA for, is to look at the expression level of these proteins. And we can do this. So there's a whole list of papers published here for different cancer biomarkers, and using, sometimes we use liquid biopsies and qPCR to look at this, but ELISA is kind of more of a quick and dirty way to look at it. It is not a diagnostic. It's not as diagnostic as it would be with qPCR, of course, but this is definitely a easier and faster method to identify some of these biomarkers and then go on and do further testing, right? So here's some other ones that you could take a look at. Okay, so any questions on that cancer bit? I'm going to look in the chat and just make sure we've got anything. qPCR. So qPCR is real-time PCR. So this is confusing because I don't have a good slide on this. I'm sorry. I do in many other slide decks, but not in this one. So real-time PCR, if we think of COVID, coronavirus, SARS-CoV-2 has an RNA genome. And when we use PCR, it's polymerase chain reaction, and that's when we make lots of copies of DNA. Well, to do RNA, we have to first convert that RNA into DNA, then we can do PCR. So that is called reverse transcriptase PCR, which is also RT-PCR, right? And so there's two kinds of RT-PCR. So reverse transcriptase is taking RNA, making cDNA from that, and then doing PCR. That's reverse transcriptase PCR. And then there's real-time PCR. In real-time PCR, we can start with RNA or DNA. So you can have RT-PCR, or just regular RT-PCR, real-time PCR. And that's when we take a sample and we're putting it in a thermal cycler that gives us a graph in real time of how many copies of that DNA we've made. And so if you think about, we're doubling the number of copies of DNA we have at every cycle And this is DNA replication in a tube. So every cycle, we're going to double the amount that we have. And you'll see a graph and it just goes up like that, right? And so we can kind of see what we're getting as it goes along. This is, if you remember doing all the PCR tests during COVID, or when we were talking about that in the news all the time, those were almost always RT-PCR, meaning real-time PCR, and also reverse transcriptase PCR. And so because there's that confusion between RT real-time and RT reverse transcriptase, we're kind of moving towards calling real-time PCR qPCR for quantitative PCR, okay? There we go. That was a really long explanation to get to what someone already answered eloquently in the chat. So, okay, now let's take a look here at immunoassays. Whoops, I went one ahead. Let me go back one. Okay, so immunoassays are a way that we, and you have probably been exposed and used many of these in your lifetime, right? So if you've ever been to the doctor for a strep test, right, and they've swabbed the back of your throat, or we've all done this with COVID, we've done this little, this one right here where you get the nose swab or the throat swab and put it in the buffer, dot it on here. Pregnancy tests or immunoassays. Drug tests or immunoassays. So immunoassays are just tests that rely on antibodies and antigens to give us an output, right? That's it. So ELISA is a type of immunoassay, and we use this for all kinds of things. So disease diagnosis, hormone detection, drug detection, GMO detection. You can actually go out in the field with a little ELISA kit to detect genetically engineered soybeans, for example. You crush them up, put them in some buffer, put your little dipstick in, kind of like a pregnancy test, pull it out, look. Are your soybeans, pregnant, just genetically modified, I should say, or not? And cancer biomarker detection, that's what we're talking about today. This is how we might test for peanuts. So allergies, eggs, things like that, pollen, right? And of course, we use it all the time in research just to see if a protein is present and also tell us how much of a protein might be present. So if we're backing up a little bit and looking at kind of the immune system as a whole, we have to talk about antigens and epitopes and antibodies and all that stuff. And so antigens are just a little piece of a foreign substance, or it doesn't even have to be foreign. In the case of biomarkers, we're looking at things that our own body is making, right? So a little piece of that. And that's what an antibody might bind to, right? So antibodies are very specific to different antigens, and particularly to the epitope, that area of an antigen, okay? Antibody structure, again, this is kind of higher level, so we're not going into a deep dive on antibody structure or the whole immune system here, just kind of telling you the basics of what you need to know. So if an antibody is kind of like a Y, right, and it's got its hands up, the hands up here, if I'm a antibody, my hands will be the antigen binding site, and these are very specific and very different. They will look different for every kind of antigen, right, and they will bind to antigens to inactivate that and call in the rest of the immune system basically, and that's our variable region. The rest of the antibody is a constant region. Okay, and so that's all the same for that same class of antibody. So antigens are kind of like these tiny flags on the surfaces of different proteins or different invaders, right? Different foreign harmful things, pollen, bacteria, viruses, right? Different toxins even. And so you can imagine them as little wanted posters that our immune system is going to use to identify, hey, this is a troublemaker, we need to take care of that. And when your immune system spots those flags, it knows something doesn't belong and it kind of gears up from action, right? And so our antibodies are basically these superheroes that are always going along looking for these little wanted posters and saying, aha, I see you, I've got you, and I am going to, now bind and things are going to happen, okay? So antibodies can bind to antigens like we showed earlier. They can also bind to other antibodies, right? And again, an antigen, in our case, we're going to be talking about with biomarkers, it can be a protein, a hormone, right? A HER2 in this case, right? So here we have an antigen with a primary antibody just binding to it. But also we can have another type of antibody called a secondary antibody that binds to a primary antibody. Okay, those secondary antibodies, you can notice they're not specific to this antigen. So we might have a primary antibody that is anti-HER2 in this case, right? And it binds. And we might have secondary antibodies that just bind to this primary antibody that it doesn't matter what's down here, what antigen is down here. Okay, why is that helpful? So to do that, we've got to take a closer look at how ELISAs work. Now, antibodies and antigens and proteins, those are all clear. If we put a bunch of that stuff in a buffer, we can't see anything. We need a way to detect and see, do we have HER2, more HER2 than we should, right? Like that was being a normal, typical, situation. Or not. We need a signal. And so we use labeled antibodies to detect our target, right? So it's detecting our target through this antibody. And this lab, we have an antibody conjugated, biochemically. So this is a job for a biochemist to an enzyme called horseradish peroxidase. This is one that's often used, HRP. And the substrate for this is something called TMB, or tetramethylbenzidine. I would never remember that if it wasn't written on this slide. So TMB. And when that substrate binds the enzyme, there will be a blue colored product that's generated. And so our well that we're looking at will turn blue. We'll just see this blue color. It'll go from clear to blue. Okay, so that's really a nice way to visualize things. And sometimes in an ELISA, they'll go ahead and add a stop solution that's just an acidic solution at the very end, and that'll turn things yellow. Okay, so sometimes if you see an ELISA, it might be yellow instead of blue, but same principle. Now, this is ELISAs happen in this little micro well plate. So it's a 12 wells, right? And you can have many of those in a little plate that kind of is about this big. They all clip into there. I should have brought one up here. I don't have one upstairs. But you're doing this in a very small little kind of plastic cup, I would say, and there's 12 of those in a line. And so if we're looking at one of these little wells, the little cups here, we'll have our antigen, our primary, our antibody that's linked to the enzyme, our enzyme-linked antibody. And then when we add the substrate, it's going to turn it blue. Okay, so that's how that works. So ELISA is an acronym actually for enzyme-linked immunosorbent assay. And now you probably know all the pieces and parts to why it's called that. So of course, we're dealing with antibodies and antigens. Those are the immuno part of the equation. Those antigens, the proteins that we're looking for, are adsorbed to the plastic wells. So they are bound to those plastic wells. I'll talk about that in a little bit. So that's the sorbent part. And of course, the assay is we're looking for a color change here. Okay, so that's the whole thing. Now, the primary antibodies. So this is a question students always make. How do we make primary antibodies, right? And this is really actually pretty easy if you happen to have access to some good, somewhat purified antigen or protein. In this case, we would use HER2, right? We're just going to inject that into an animal, right? And we would want to use something that's different from a human. So a rabbit is often a good thing, mice, goats, there's all kinds of animals that we could use. When we inject that HER2 into the animal, into the rabbit, that bunny's going to be like, hey, I don't recognize that stuff. I'm going to make antibodies against it, right? So it will make lots and lots of antibodies against that protein that we've injected there, right? If we were looking for COVID, we would inject COVID antigens here, right? If we're looking for HIV, we would inject HIV antigens. And so over a course of a couple of weeks, we're going to inject the bunny multiple times with these, get its immune system going, take a blood donation from the bunny, and now we have these rabbit anti-HER2 antibodies in our case. Okay, those are our primary antibodies. Now, in this lab, we're going to be using primary antibodies and secondary antibodies. So I'm going to explain how we make secondary antibodies, and I'll explain why we're using those in a minute. Okay, so secondary antibodies, those are antibodies against a primary antibody. We have rabbit anti-HER2 antibodies that we've already made. Now we want to make secondary antibodies. We're going to take those antibodies, those rabbit anti-HER2 antibodies, and immunize a different animal with them. We don't want to use a bunny again. We want to use a different animal. So let's use a goat. Okay, so inject that goat with the rabbit anti-HER2 antibodies. Do it several times over the next few weeks. And now the goat's going to be like, I don't know what this is, all these rabbit antibodies. That's not right. That's not goat stuff. I'm going to make antibodies against those. So the goat cranks out antibodies. We get a blood donation from the goat. And now we have goat anti-rabbit antibodies. I want to make a distinction here. This goat antibody has nothing to do with the HER2 or whatever antigen we originally injected into the rabbit, right? It's only really made against the constant region of that rabbit antibody. Okay, so it's a little bit different, right? It's not going to directly detect our antigen that we're interested in detecting, HER2 in our case. It will just detect anything that's stuck to it, the antibodies that are stuck to it. Okay, So why would we use secondary antibodies? What's the point? Well, they're cheaper, okay? And this is why. They're more versatile, I should say. So if we wanted to, we've got our goat anti-HER2. Now let's say, I want to look for a different cancer biomarker. I'm going to look for that EGFR2. I'm going to inject that into a bunny now. Okay, so here's my other cancer biomarker. We've injected that into a bunny. Bunny is going to make antibodies against that. They're different from the first primary antibodies, right? So here's our rabbit anti-EGFR2. Did I get that right? Yes, EGFR. Okay, so our other cancer biomarker. Now, here's the cool thing. Since that goat anti-rabbit antibody is not specific to the antigen that we're detecting or the protein, the hormone that we're detecting, we can use that same goat anti-rabbit antibody to detect this new rabbit anti-EGFR antibody. Okay, I'm going to let that sink in for a little bit because it's a lot of, I feel like I've said the word antibody like 40 times here. Okay, so we're able to use the same goat anti-rabbit antibody to detect multiple types of rabbit anti-whatever antibody. Okay, I could use it, I could inject COVID and it would detect that as well, right? So it gives us that versatility, okay? And here's why that's important. I'm going to come back to, I don't want to get too in the weeds on monoclonal and monoclonal. These are polyclonal antibodies that we're looking at here. I'll talk about why we're going to do why we typically use primary and secondary. So there are different types of ELISAs, and the first easiest type is direct. I'm going to show you what that looks like. The second kind, the kind that we're going to talk about today, is indirect, and so it's using the primary and the secondary antibodies, right? There's other kinds too. Sandwich is one that we commonly use where we have an antibody and then we add our sample and then we have another antibody that detects that. So it's kind of, you're sandwiching the sample inside between two antibodies. And we have competitive ELISAs. This is what we typically use for drug detection. Okay. Now, direct and indirect. We eventually want to get to indirect. I'm just going to start off with direct just to kind of, it's a little bit easier. Okay, so we're going to start there. So ELISA, when we're detecting antigens, so, we would take a sample here. It might be blood, plasma, saliva, urine, for breast cancer, it will be a blood sample typically. And we're going to add that into our 96 well plate, or one strip of it. That's what your students will do. And if we look at a well, just one of those little circles, right? What's going to happen? Add our sample. Proteins are going to bind to the well. We'll add our antibodies next. Wash out anything that's not bound. And then we can, in our case, we'll add our secondary antibody, and then we'll add our substrate. And then that's when we get a blue color here. Okay, so that's how we would detect antigens. I'm going to skip past antibodies because we're not going to do that today. I'm going to skip back to this too because we'll come back to this. Sorry, I should have done that. We have a whole video on how to do the lab that I'm going to show you. I don't want to go through it now, but that is there. So let's take a look at the details of how an ELISA actually works. So usually we do these in triplicate. And so each of these little strips, so if we're looking at one row all the way across, it's a separate strip, right? And there will be 12 wells in there. And so students are going to label these plus, 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 minus, 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 1, 1, 1, 2, 2, 2, or AA, BBB, doesn't matter, right? And if we look at each well, we're going to talk about what's happening in each well next. So actually, I'm just going to skip ahead to an indirect ELISA. There we go. So let's look at an indirect ELISA, which is the one that we're going to be using. So the first thing that happens, they're going to add their patient sample to the well. So this would be a patient from, potential breast cancer patient, for example, right? And they're going to add that sample to the wells. Any proteins in that sample are going to bind non-specifically to this plastic well, okay? And so this is the one we're looking at, this green one. That's our HER2 that we're looking for, okay? There's going to be other gunk in there. They're going to bind as well, but our antibodies aren't going to detect those, so that's okay. All right, so that's the first step, adding our sample. Now we're going to do something called blocking. So we're going to add a wash buffer. And the wash buffer serves two purposes. It's going to wash out any unbound antigens. So you saw that this guy was floating up here. And now when we add the wash buffer, he's magically gone, right? So we're just you add it and you dump it out, add it and dump it out, 2 washes. the other purpose of the wash buffer is to coat all of the uncovered surfaces of the wells with, it's got some detergent in it. And so that detergent will coat the surface of the internal surface of that well, so that now when we add our antibodies, they don't stick non-specifically. Antibodies, when we add them, they can only stick to things that they would normally bind to. In our case, our antigen here, HER2, right? We don't want antibodies just binding willy-nilly here, or it'll give us a false positive. So this wash step, this blocking step, is really important. So next thing we're going to do is add our primary antibody. So primary antibody is going to bind. This is our rabbit anti-HER2 antibody. And it found HER2, right? It's going to bind. There will also be unbound antibody. We want to get rid of all that. So we're going to do another wash step here, right? So get rid of all that. The next step that we're going to do is we're going to add a secondary antibody that's going to detect our first primary antibody. So this is goat anti-rabbit antibody, right? In our example that we did. This is what our enzyme is conjugated to, right? So our enzyme is conjugated to that secondary antibody. And so it will bind wherever there is a primary antibody that was made against. So if there's any rabbit antibodies floating around in here, this goat anti-rabbit antibody is going to bind. What are we going to do next? Wash everything out that doesn't bind, right? So I'm going to get rid of everything that's not specifically bound. Okay. Now we're going to add the substrate, and the substrate will cause this color change. And we can see that we indeed have, our HER2 molecule that we were looking for here. Okay, so those are the steps of an indirect ELISA. Now, I want to talk a little bit about how all these steps happen. So in that first step, when the proteins absorb to the plastic, why does that happen? Why does the proteins just stick to the wells? It's just hydrophobic interactions, and it's a pretty strong interaction. People have asked, can we reuse the wells? And kind of, but it's a pretty, like I said, you'd have to really get after it with soap and stuff. It's been done, but the strips are pretty cheap, so I probably wouldn't recommend it. But any kind of protein is going to stick there, just non-specifically. Antibodies will stick, antigens will stick, whatever you happen to have in your sample is going to stick. The wash buffer, two purposes. It's going to wash out anything that's not stuck on there. And it's also going to coat the exposed surfaces of the wells that aren't already covered by proteins from our sample, so that our antibodies that we add in the next step don't stick non-specifically. We don't want those sticking non-specifically. We only want our antibodies sticking to the thing we're trying to detect, which is HER2 in this case. Okay. Secondary antibodies. Why do we use these? Could we just attach that enzyme, that horseradish peroxidase, to our primary? Absolutely, we definitely could. The reason that we tend to use secondary antibodies, there's two reasons. So take a look at the difference here between our signal output. And I will say that I just didn't have room when I was doing this. There will be a lot more of these secondary antibodies attached to each of these primary antibodies. You might have, tens attached, right? And again, they're binding to that constant region of the antibody all over it. So there's more room for them to bind to. So what we're looking at here is we have now this signal amplification, whereas before it was kind of a one antigen to one antibody situation if we were just using the enzyme linked primary antibody. Now we have, for every one antigen, we might have 5 times the amount of signal output from the enzymes that are attached to all those secondary antibodies, right? So hopefully that makes sense. That signal amplification, that's a big one. The other reason is it's cheaper. Okay, so like I said earlier, we can use that same goat anti-rabbit secondary that's enzyme linked to detect multiple different things. I can use that same secondary antibody to detect HER2 and EGFR, right? If those antibodies were made in rabbits. So it's cost savings. Okay, that act of putting the enzyme onto an antibody, that's expensive, right? There's a cost associated with all of this, but we want to minimize the cost as much as possible. Okay. Now, if you want your students to go through and look at these steps one by one, we have this paper model and it is included in the PowerPoint. It's actually not the last slide. I moved it up to the top. I'll have to fix that. And I'll just kind of quickly go over this. They're basically pattern matching. So here's an antibody. And you can see these are our antigens. So if this is HER2, we try to make it where you can print this in black and white, right? And so you can look for some different patterns. But you can see the solid black on this antibody, the variable region matches the solid black antigen. Okay, so this is a match. And now the secondary antibodies won't match. They're the wrong shape. So you're matching pattern and shape, right? So they have this pointed edge versus the squared off end. The secondary antibodies are actually going to match on the little dotted areas of the primary antibody. So you're going to overlay them just like this, right? And you could actually overlay several of them onto, this has spots for 4 different secondary antibodies that you can put on this one primary antibody. So they can cut these out and build this as you're going through that slide deck with them. They have their antigen down, right? They can just use a piece of paper as their well. They'll put their antigen down. And then that's adding the patient sample, their antigen is going to go in the well. They can wash it out. We even have little drops of them to, mimic washing them out. Then we add our primary antibody. They could add the primary antibody and have it bind right on the antigen like this. Wash out everything that's not bound. Add the secondary antibodies. So we have something that looks like this, right? And so it's going to look at the end of it like this. So here's our primary. We're going to wash out things that don't bind. Here's our secondary, wash out things that don't bind. And here's our substrate. Okay, so they can do that right alongside you. It works really great if you are able to laminate these and cut them out, save them from year to year. The other thing is you can have your students do the protocol straight from this modeling activity. So on the back of the pieces, if you've laminated them, you can tell them how much of each of these they're going to be adding. Right. And so I'll get to the protocol in a minute and write the incubation steps. Then when they go to do the lab, they can just flip over their model pieces and they have the whole protocol written just right there. Okay, so pretty easy. Sorry, this is the antibody. We have both of them in here. I want to skip to the activity protocol. So there are a few tips here that I want to mention. Cross-contamination is going to be your big issue in the classroom. And it's because students just don't realize how important it is to keep things super sterile and avoid touching one thing and then touching another. So they're going to be working with either micropipettes with tips or with disposable pipettes. Either way, if they're working with something and they touch that plastic tip, either the micropipette tip or the disposable plastic pipette tip, to something, they need to change it. Okay. If they've touched the well or they've touched liquid in another thing, they do not want to transfer that over to another well, or they will cross-contaminate everything. This is one reason we do things in triplicate, is because sometimes if you get a little cross-contamination, you can maybe see maybe the other 2 wells are okay. But ideally, just reinforce with them, if you touch anything, just change tips. Right? Not hard. Tips are super cheap. And the other thing that they might do is contaminate their wash buffer. Remember that wash buffer is a step that we add. It's buffer that we add in between each of these steps to wash out things that aren't bound. And so sometimes what they'll do is they'll add that wash buffer to a well and touch a pipette into the well and then put that pipette back into their wash buffer. Now their whole wash buffer is contaminated. So because of that, I don't give them a ton of wash buffer, maybe 50 mils, hold some back. And I'm just like, hey, look, it happens a lot. Don't worry about it. If you accidentally contaminate your wash buffer, just let me know. I've got more, right? So don't make them feel bad for it or anything. Yes.
So hi. So adding to the information on the wash buffer, we have a question. Yes. Would it be incorrect to refer to wash buffer as wash blocking buffer?
So I do hear people call it blocking buffer. In this lab, in our protocol, we call it wash buffer, but I don't think it's incorrect to call it blocking buffer. Yeah, and people use different things for blocking as well. This wash buffer just happens to do both purposes. It's got that detergent that washes the wells out and also coats the wells. So, but people use other types of blocking buffer as well. Yeah, great question. They're also going to be tapping their, when they're getting rid of the liquids in to put the next liquids, in the wells, they'll be dumping it out onto paper towel. I'll show you what that looks like. And we want to make sure that they're moving it between every time that they kind of tap it on the paper towel. So here's the procedure. I'm going to walk through it really quickly. It's pretty easy. We're going to look at a quantitative ELISA. And so this one, in this lab, we're seeing, again, we're all expressing HER2, right? So just seeing yes or no isn't enough. We want to know, is there an overexpression of HER2, of that transmembrane protein or not? So you would, I don't know the actual concentrations, but there are probably papers on this of what would be considered an overexpression of HER2. And they are going to set up a serial dilution of a known quantity so they can compare it to their patient sample. So they're going to have two strips. One of the strips they'll label 1 through 12, and the concentration they'll just write in their lab notebook. They're going to make this. The second strip they'll label, this is kind of our test strip, right? And so they'll have 3 wells that are plus, 3 wells that are for the negative control, this positive control, negative control, 3 wells for patient A, 3 wells for patient B. Okay, so something like that. To set up their serial dilution, what they're going to do is they're going to start with this known quantity, 1,000, that should be nanograms, not nanometers, nanograms per milliliter of antigen, and put it into the first well. And then in the rest of the wells, 2 through 12, they're going to add 50 microliters of PBS. If you're doing serial dilutions, you would not want to do the disposable pipettes here. If you can, at all possible, use a adjustable volume pipette, right? Because we're looking at really precise measurements here. Okay.
Lee, I just want
Yep.
to chime in because it's probably a really good question for the
Yeah.
audience in general. Can we get the HER2 antigen in the kit?
So the HER2 antigen, that's a great question. The HER2 antigen doesn't come in the kit. And actually the antigen that comes in the kit is just, what is it, Tamika? It's egg stuff. It's egg protein, right? And so,
Right.
but you can call it whatever, right? We've actually thought about maybe we should do like a food allergy test with this, right? So the whole idea here is that serves as a positive control for you to wrap your own storyline around. And it can be any of these storylines that we've talked about. Great question. Can you get HER2 detection ELISAs? Yeah, you can, and you can get antibodies for that stuff. But you don't need to do all that, right? So we're just kind of going through this with the students. It is actually antigens and antibodies in the kit that they're using though. I think it's chicken gamma globulin, and then it's either rabbit or goat primary and secondary. I can't remember which order they are. It does say it in the kit though. Okay. So they're going to have their concentrated sample in lane 1, and that'll be full strength, 1,000 nanograms per milliliter. The other wells will have PBS. This is just a buffer. And it's the buffer that we use to make the wash buffer and to dilute the antigens in and the antibodies in and all that. And then they'll do a serial dilution. And so what happens with the serial dilution, you can use the same tip all the way through, but they're going to take 50 microliters from this first concentrated one and add it to well 2, go up and down with their pipette to mix, take 50 microliters out, add to well 3, so on and so forth. Well 12, we want 0 in there. And so you're not going to take 50 from 11 and put in 12. You're just going to discard it. Okay. So now this is set up with we know the concentration of all of the antigens, all the proteins. In our case, we're going to call them HER2, right? In each of these wells. And it's going to go from dark here on the left all the way to light blue and clear on the very right. Okay, so they're setting up their serial dilutions now. Now they're going to be doing the same thing for both of these. So we're going to be on the second microstrip plate, we need to add our proteins to these wells, right? We add it to the serial dilution strip. We want to add it to our test strip too. So they're going to, in the kit, we have positive control antigen, right? So they'll add positive control here. They'll add negative control here. This is just buffer, no antigen in it. They'll add their patient sample here. You as an instructor would have set this up to whatever concentration you want, right? So in this example, you'll see it's about, let me go back and look. It's about 31-ish nanograms per milliliter, but you can make it whatever concentration you want, okay? For A and B, A might be a lower, B might be a higher, A might have some, B might have none. That's all up to you, right? Whatever storyline you want to make there. They'll incubate this for 5 minutes. The 5-minute incubation is not entirely necessary. I would say 2 to 5 minutes is what we want here. Okay. And I've got the paper model over here on the right. So you can kind of look at, they could be doing that along with this. This is what's going on inside the wells, right? Okay. So 5 minutes incubation for both of those strips. Now everything's going to be the same for both of the strips. So, and again, the reason that we had it different is our standard strip is just our serial dilution, and then we have our test strip. But what we added in both of those were antigens, the antigens that we're looking for, in our case, HER2, okay? we want to dump out both of the liquids onto a paper towel. This is the only thing that I found those brown school paper towels to be useful for, is about like 1 ml of fluid. So you're going to dump that out, and they need a big stack of these. So they're going to dump them out and tap, tap, tap. And they want to move it every time they tap, because we don't want water or the liquids splashing back up into the wells, right? So just, be generous, let them go nuts with the brown paper towels. And they're going to do that washed up twice. Okay. The next step is they're going to add primary antibody all the way across both wells. So now, again, if the antigen's there, our primary antibody is going to stick. Okay. So if we're looking at our, we've got antibody, we've got antigen in all these ways except for 12. So we would expect to see primary antibody sticking here. In these, we've got antigen in the positive controls and maybe in A and B, right? But not in the negative controls. Incubate 5 minutes, dump it out, wash 2 times, just like we did before. There's 2 washes. I don't think I mentioned that before. Next step, and you might remember this from earlier, secondary, our enzyme-linked secondary antibody to every well. Sorry, let's just say secondary. It could be enzyme-linked and secondary, same kind of thing. It's our secondary in this case. Incubate 2 to 5 minutes. Next step, wash it out. And in this case, wash it 3 times, okay? And we want to make really sure that we don't have any rando secondary just hanging around there. Okay, so 3 washes here. And then finally, we're going to add the substrate all the way across on both wells. It'll start changing color almost immediately, especially if you've waited 5 minutes for the other ones. If you've waited 2 minutes, it'll take a little bit longer to change color in the back end, but again, not more than a couple of minutes here. So you'll start seeing this blue color develop. Okay. Also, I wanted to mention the micropipettes versus disposable pipettes. Since this is quantitative, just adjustable volume pipettes are ideal. If you don't have those, though, you can set up the standards for them, or you can set up one standard. You're probably going to need one pipette to do this for you. And then they can use their squeezy bulb disposable pipettes and just add 2 drops instead of 50 microliters. So everywhere it says 50 microliters, they would just add 2 drops of liquid. Okay. ELISA is one of those things where we just want to get the surface wet, right? We're not really, we're trying to kind of get everything into the wells, but the amount doesn't matter as much. Quantitative, it matters a little bit more. So if we're doing the yes and no one, definitely go ahead and do the disposable. For this one, if you really want them to get true quantitative results, stick with the micropipettes. But if you don't have them, the disposable pipettes will work as well. And then they're going to visually compare and see what they got. So if they have a microplate reader, that's great. They can pop them in there. You don't need one, though. This can just be an eyeball thing. So here we go. I'm sorry. There we go. So they're going to get something that looks like this, right? So here's our standard, highest concentration on the left, all the way down on the right. Here's our test strip. We've got positive, looks good. A, they can visually compare here and say that A looks kind of similar to maybe 6, 5, 6, 7, somewhere in here, right? So maybe it's between 16 and 63 nanograms per milliliter. If they have a microplate reader, they can pop that whole little tray in there and get a reading from that. There are also apps where you can do this too and get colorimeters and things like that. It's a little bit trickier, but the microplate reader works really well. And they can visually compare, and they can even make a standard curve if you want to get into graphing on the semi-log paper here. And that is basically it. If there are problems, if you see everything turn blue all the way across, something's probably contaminated, likely their wash buffer. Okay, so then maybe they didn't change tips. They also could have just not been paying attention and put positive control all the way across on that first step instead of in the first 3 wells. Things happen, right? So if this is only for one student, that's probably what happened, one of those 2 things. If your whole class looks like this, I hate to tell you, it's probably something you did in the prep, okay? But more likely what you're going to see if you have a prep problem is nothing turns blue. And the biggest reason that you might see that is because when you're diluting the the antibodies, if you add wash buffer to it, it won't work. So you have to add PBS to that first primary antibody instead of wash buffer. It's very clear in the directions, and in this PowerPoint, we have it in there as well. But if you accidentally do that, you'll get no results, and that's kind of a bummer. A lot of times that we'll see is something like this, where students have kind of their negative controls or maybe a little bit light blue, and that's probably from things either spilling over or they touched a tip inside the well or something like that. And so just depending on how it looks. If it's like maybe one that's blue and the others are clear, you can probably go ahead and interpret those results and explain this is why we do things in triplicate, right? So you can kind of get some results out of that. And I think that is it.